So it is epigenetics, epigenomics we're talking about today, and why are we doing that? Why epigenetics, epigenomics? An interesting field. Well, the deciphering of the human genome sequence has of course created a new area in the understanding of biological processes and in both health and disease. However, and that's important, the way in which genomic information is organized in the cell through epigenetic processes is known to play a major role in regulating gene expression and in controlling cellular functions. So, the epigenetics and epigenomics research explores those processes, the processes that go beyond the DNA stored information, are essential for packaging and interpreting the genome, fundamental to normal development and differentiation, and increasingly being recognized as involved in human disease. So, what is then epigenomics? Well, it should be said on this, but I should say on the Mac computer is all right, but when it's turned over to Microsoft, and had a witness for that too actually, that it is. And for reasons I don't know, but it may have with an old conflict between Apple and Macintosh so that the programs are didn't solve. Anyway, what it says, it should say here.
That tells us epigenetics, we don't know anything.
That's probably the Microsoft, yes, true. The Apple knows what it is. Or, no, it says that literally epigenetics means outside genetics or upon genetics. And is has more specifically been defined as the study of mitotically and or meiotically hereditable changes in gene function that is not due to changes in the genome sequence. Or more simply, the study of non-DNA sequence related heredity. When was epigenetics established, or the word epigenetics? And that was in the 1940s by Wellington. He coined the term epigenetics, and he did it meaning the interaction of genes with their environment which bring the phenotype into being. In the 70s, when the molecular genetics, molecular biology has started up, one could go and look a little bit further, as you know, into the cellular molecular mechanisms, and Pope and Holiday, they know, of course, about the Wellington, and they noticed that when genes were off in some, that they were modified by DNA methylation. So they say that the molecular mechanism of epigenetics was based on the covalent chemical modifications by methylation of cytosines in the CPG denucleotides, meaning that you have the C5 prime, the phosphate part of the G. So it's not GC, it's only the C's when they are in CG. Right. All right, so if you now look at the major epigenetics mechanism, it's more than DNA methylation. It's certainly one, and important. But you also have histone modifications and various means for chromatin remodeling. And you have the RNAs, microRNAs and the long non-coding RNAs. So, if you should look at a little bit of an overview here, genome function and cellular phenotypes are influenced by DNA methylation and by protein DNA complexes known as the chromatin. Now, DNA methylation occurs on the cytosines, as we said, the 5' end of guanine. And the accessibility there has to do with that the histone binds to. You know that it's getting wound around the histones as the nucleosomes, and there are also some other DNA binding proteins, and the histones are critical in which type of modification they show. They could be acetylated, methylated, or phosphatated. And also, there are this polycomb complex of protein which associate with this chromatin. And in addition, which are mostly of a repression type when it's activated. And then in addition to that, you have the non-coding RNA, which represents an additional layer of chromatin structure that affect genome function in a context-dependent manner. As an overview of how the DNA, simple picture of how the DNA regulate or is involved, say, in the gene regulation is an active gene. Here you have the promoter, the promoter tata box promoters and enhancers, and here is the messenger started to be. The methylation sites, or the CPG sites, there are only a few of them, or none, are methylated. If that is the case, the gene will be active. If the CPG sites, mostly over the region here, in promoters and enhancers, and the larger parts of that, is mainly methylated, the gene will be inactive. So, how comes the DNA methylation about? Well, it is that that a methyl group is associated with the 5' or the 5 position in the cytosine, and it is in the CPG direction most of the time. But there are cases, evidently, where there are CPA, CPT, CPCs also, although that's much more unclear. And a study, now 10 years ago, when was it? Yes, it was 9 or 8, 7 years, showed that it seemed that a lot of people were working, trying to find it in stem cells, in human stem cells. There's in vitro cells, of course, but anyway, up to 25% may be non-CPGs. Whereas in, for example, a standard human fibroblast used for a lot of things, including making or producing the iPS-induced pluripotent stem cells. But then they have 1% of it. And as it seems, continuously in the adult, there is a very, very little, but during the embryonal development, there may be a higher percentage of non-CPTs. Whatever. Now, the methylation machinery, that consists of two types of components, the DNA methyltransferases and methyl binding proteins. The methyltransferases, they establish and maintain the DNA methylation pattern, and there is a distinction here. There are particular DMT1s which maintain methylation, that's the one which is most active all the time. So, and then there are the DMT3, ABNL, which is de novo methylated. And then you have the methyl binding proteins which are involved in reading the methylation marks. Oh, there it is. Oh, yeah. So it could be, DNA methylation could, or promoter sequences can repress transcription by two mechanisms, either directly by blocking for the transcription factor to bind to the promoter, or indirectly by the methylation binding proteins are binding, of course, to the CPGs, methylated CPGs, which blocks the transcription factor binding. So, then we should take the second mechanism, and that's the histone modification. And that is a much more than nucleosomes, much more complex. Yes?
Are there as many CPGs in the gene itself?
I'll come a little bit to that. Okay. But the question is good. It has to do with its position. Now, the histone modifications. That is a much more complex situation in terms of alternatives. As we said previously, and you see here in the nucleosome, the N-terminal tails of several of the amino acids in the histones, they may be modified. They may be acetylated, methylated, or phosphorylated. And that makes a difference to their activity. As again, a simplified picture here, you could say that the epigenetic modifications, when the histones are acetylated, the DNA is not methylated. That means an open configuration, which means that the gene is active, or that sequence is active. If it's deacetylated by methylation, or by methylation usually, and the DNA also is methylated, you have the histones, the H1 histones, we're keeping together in a more tight structure, the whole genome sequence there, or the whole chromatin sequence, which means that it's closed and cannot be transcribed. It's the gene function, it cannot be transcribed. Now, if we look at the various types you can do, the various ways that histones can be modified, the different amino acids, and lysine, and I don't remember the name. But anyway, it could be 1, it could be 2, it could be actually 3 methylation or methylated groups. And it could be, you see on some of them, this, those of you who know the code for a figure of an amino acid, you see all of these here, and they could be methylated, acylated, ruled up, right? And it has been estimated that there are more than 400 different types of modifications of combination. And you realize, of course, that trying to figure out which specifically is of importance in this given situation is very, very difficult. It's easy to start counting the stars, almost. Luckily, there seems to be a mutual reinforcement between histone and DNA methylation in the inactive group. That is, if DNA is methylated, they're signaling through other molecules like HERDAC that histone will be deacetylated. So, and the vice versa. If histone is deacetylated, it will signal demethylation with that, and it will then recruit DNA methylases, methyltransferases, and the DNA will be methylated. So, going at the DNA methylation, which we mostly do since it's a fairly simple, clear-cut way of doing it, it tells us quite a lot of what is the rest. So it gives a good picture also of the histone. In general, of course, there are no. Now, looking at the full picture here of an active or an inactive gene, there is a lot of proteins, both when it's active around the sequence of the gene, whether it's active as here or inactive. But there is a distinction between which proteins are there if the promoter is not methylated. As here, you see, over the exons and the promoter here, there are a number. If it's methylated, there are a number of others. Oh. And then, of course, comes the microRNA and actually non-coding RNAs. The microRNA, they are there and they have a role, but we know in principle that they could act by regulating gene expression in 2 ways, by base pairing. If they base pair to an homologous sequence of RNA that is a messenger RNA, it will target that and it will cleave the messenger, so the action of that gene is up. But also at the transcriptional level, the RNA-specific microRNA can target the homologous sequence on the DNA base. And that is in one particular gene and only there. And that will cause that the target DNA sequence will be methylated. And histone modification will come on. And that's done, and I have done it myself, some studies of that long ago. That is true. You could see that the promoter regions are getting methylated if you're using the micro or cRNA, which is principal type. So, but to what extent then, how it's sort of coming to the picture in gene regulation really is a little bit uncertain, or quite uncertain. It has a role, no doubt, but more specifically how and when. So let's take a look here at the changes in DNA methylation during the developmental stages. There are two major reprograms. We talk about the reprogram in the early embryo and the reprogram in the germ cells. So if we start here in the early primordial germ cells, when you know the gonadal stage of differentiation, it starts in males to produce the sperms. And that goes to the initial phases comes very early in your embryonic development. And in female, of course, it's in leading up to what will be eggs. And they are, the sperms in particular, quite a lot of DNA methylation, DNA methylated. So the eggs a little bit less. Once it has first light and the zygote is formed, a process of demethylation in the early embryo goes on. During the stage of morula down to the early blastocyst, that is actually only cell divisions, by cell division without having any kind of differentiation. It's the same cell which is multiplied. However, when you come to this stage of blastocyst here and development into the various types of somatic lineages come up, then the DNA methylation starts up rather heavily and differently in the different parts of the images here. Up to the adults, that's where it is kept and kept on by maintenance methylation in principle. Adult state. But you have also the de novo methylation, it's a separate product in the trophoblast lineage, leading to the placenta, yolk sac, and so forth. And of course, there are these few cells which also should rise to the primordial germ cells, which, as you saw coming in here, when they reach this stage, they will methylate again, right? So you have the methylation of the germ cells here and of the early embryo here. And this is repeated all the time.
So,
Yep.
go back to that.
So,
The epigenetic changes that are inherited are passed from generation to generation.
mhm.
Are they controlled by other epigenetic phenomena besides DNA methylation? Because I mean, you're removing it and then putting it back.
Yeah, they probably are, but we know that DNA methylation, or these changes, are not as strict. It fails sometimes, and it can be re-established, but it is at a much lower rate than its mutation. If you got a long DNA base, it is in 999 cases of a million. So it's one in a million which goes through. The rest is repaired. When it comes to CPG, or the methylation here, it's 1 in 10,000. So it's 100 times more frequent that there are these changes. If they go further, they may cause it, of course, depending on a little bit where they are established. But then there are particular mechanisms also about that. So there are disturbances in this methylation pattern. And I come to it when it comes to imprinting, because the imprinting diseases are related to that. There are disturbances in that. Anyway, I'll just take a shortcut here on the CPGs. As Roger asked about, the CPGs are mainly found in what we call CPG islands. And that is stretches of DNA which are 500 up to 1,000 bases. And they usually contain over, if you look at them, more than 60% of CPG nucleotides, CGs in the nucleotides sequence. And the limit is that 55, it should be over 55% to be classified as a CPG island. The CGIs are often located at the 5' ends of the gene, and particularly in the promoter region. And in housekeeping gene promoters, they are usually unmethylated. And keeping genes, what is that? Well, it's the genes keeping the cell going as a cell. The basic mechanisms of being a cell. The genes setting that up, they are called housekeeping genes. Whereas the tissue-specific gene promoters, of course, they are methylated when they are not active in the tissue. And since we know that only 10 to 20% of the genes is active in any cell. All cells have the same DNA. but 10 to 20% of the cells are active. 80 to 90% are not active in particular cells. I skipped the figure on the CpG number on humans. I'm sure it has changed when I started looking at it. I realized that I haven't looked for the figure now. Anyway, it seems that it's much more in humans than it is in the mouse. And I think that today, when you want to know a little bit more about the CpG islands and their status and where they are and so forth, you go into these genome browsers, you look for the CpG islands over a certain region, and you can see where you have the exons, you can see where you have other types of markers, you can see where they have the CpGs, and you could go to the individual level. So this is the way you sort of sort out if you're interested in the given region. Now, it seems that a few years ago, well, actually it was also in 9 years, it seemed that it's not the CpG island which is the most critical one. It seems that it is what is called the CpG island shores. Here is a gene, and you have the CpG island, and here is the CpG island shore. If you look, the individual dot series for individual for different individuals, this is human, this, and you look for 3 tissues: brain, liver, and spleen. And you see here that here it's the average signal, sort of methylation, is about the same here. But here, it's a great difference in this, right? And if you look at this one here, you have the same tissues normal, but also you have the colon normal and a tumor. And there you see it even more clearly. The green here is a normal colon in the shore. Fairly high degree of methylation in this particular gene, CpG shore, whereas that in the tumor is much lower, that is heavily demethylated over this region, which might be one of the important mechanisms behind it. Now, here comes a question which has been there and still is. What identifies a specific genome locus for DNA methylation? What mechanism does show or point out into the cell, this should be methylated? Well, if there are various, say, speculations, and there are some basis, some data also behind it, that RNA, as I told you before, it may identify specific parts, specific sequence, 15, 20 bases, exactly there out of the 3 billion bases. It's there, nowhere else. But it cannot be proven. And we have the cyst in the inactivation, the RNA, which is long non-coding RNA involved in the X chromosome inactivation, as you know, in females. You have the Wi-Fi and SNF models in the chromatin remodeling, and you have the existence, and you know, there is a lot of retroviral sequences all over the genome, and they make some very strange interactions, and they may be induced to do that, and if they do that, may cause that there will be in these regions methylations. But we actually don't know. It's a very fundamental question, but still waiting for answers. People are looking for it. There's all over now and then. So here we have a picture then of the epigenetics in various phases of our lives, starting with the sperm and egg and the zygote, and during embryogenesis, and when we are getting adults and getting older, the senescence process here. And in the very early, already in the egg and sperm, we have a process called genomic imprinting, which leads to what we call a parent of origin inheritance. That is, a certain phenotype is dependent on if you inherited from the mother or from the father. It will be in one way if it is from the mother, and one way if it is from the father, if there are changes in. Now, and then we have the embryonic development, cell differentiation, as we just talked about. You have the X-Inactivation, which you, of course, hear a lot about. And then the question of how different environmental factors can induce epigenetic changes. We know that they do, and we'll talk a little bit about it. We have the aging, which is also related to the methylation, and then we have a lot of the aging-related diseases, common diseases, cardiovascular disease, diabetes, obesity, schizophrenia, and of course, cancer, which is involved everywhere for changes. Let's look at imprinting, or this phenomena of parent of origin inheritance. In the gametes, these imprinted genes, let's say that, there are about hundreds we know about. There could be a little more than that, and about 50 of them, we talk about maternal imprinting, and the other 50 that this is paternal. And that means that in this case shown here, there is a sperm and there is an egg. The A locus, that is active in sperms, but is methylated, inactive, in the eggs. And the vice versa. The B locus is methylated and inactive in sperms, but active in the eggs. So, in the somatic cell, you have one dose of gene A, one gene dose. And you have one gene dose of gene B. So this is a gene doses regulation, which is very important. There are a number of other genes actually, which do behave as if they are related to that, much more than we generally think of. Anyway, during spermatogenesis, let's say this is a boy. It should produce sperms. Well, the sperms should only carry the B gene methylated. The A gene should be active. So the gene he got from his father and the gene he got from his mother, this one has to be changed. Actually, both of them, or all of them, and they're putting new prints on. And the vice versa in the oocytes, right? The egg cells. So there is a process of demethylation totally, and then they put specifically in the spermatogenesis the cells which should be imprinted in males, and in the oocytes, the cells, the genes which should be inactivated by imprinting in the females. Yeah, the specificity, as I said, a little bit unclear about it. But what we know, and that I think might be of some interest, and it seems that, why do we have imprinting? Well, there's a lot of speculation, the hypothesis of various kinds actually. One effect is that it is seen, or it can be shown that it has co-evolved. Genomic imprinting and live birth has co-evolved in evolution. And it seems that it was established some 180 million years ago. So even the early marsupials, predators and primates and so forth. But the monotremes, they didn't. They had an egg type of, they were mammals, but they had an egg type of birth for the gene. Now here we have an example of a imprinted disease. It's called the Beckwith-Wiedemann syndrome, BWS. And it's quite a complex one. But still, in this case, in the female, we should say the BWS is an example. BWS is only manifested when it is inherited from the mother. And as you see here, this man, he had been a family with this woman who happens to be dead, and then he was remarried or whatever, and had some other child here. And you see, none of his children are getting the BWS, either in this or in this groupings here. However, the females here, this daughter of his, I should say, she got 7 of her kids being having this syndrome. And so did one of the daughters here get 7. These others here, either is known, I don't remember what it says, or they didn't have any. Now, the female is normally imprinted here. So if there is something wrong with the imprinting in the female, it means that in the female that there will be her. She is supposed, he leaves an active one, and she should leave an inactive one, imprinted, but she doesn't, because she has inherited a change in the imprinting. So the BVS individuals, they are getting 2 active genes instead of 1 of a given time. And that was causes, and these are related to growth and rather heavily growth in the BVS people. I don't know if you have something here. I remember we discussed it years ago, or maybe you have something. Anyway, let's move on here to, what happened now? Okay. Oh yes, we don't go so much into that. I guess you know about the X-Inactivation, which also is a dose regulator. And it should be whatever. If you're male or female, if you happen to have, as it is here in Klinefelters and so forth, there is only 1 X active. And there's the example of we could just mention, I don't know now, but there has been a lot of speculations when exactly during the embryonal development the inactivation starts, but it should be pretty early. Here's it, late blastocyst. I don't know if that's correct or if they have specified it even more.
Sorry,
You know it? You know it?
Nope. Yeah.
Sorry. Now, we should look at the environmental impact here, and with genetics. And there are a number of those we know are effective here, and it comes to nutritional supplements or symbiotic chemicals, that is those not being in nature, and you have behavioral cues and the reproductive factors. The RT is a disturbing fact here, that it does give rise to a number of changes in, which is not good of course, and low dose regulation. For reasons just of observation, it has been noticed that promoters, genes, as you say, which are targeted by the environment, seems to be promoted in housekeeping genes and also in some tissue-specific. But then there is this transposable elements, which in a lot of situations has been shown. But that's the cause, a gene which is related to transposable elements and regulatory elements in imprinted genes. We should look at one established example here, and that is in the so-called agouti mice. There is a transposable element in one of the alleles here. It's sitting about 100 kilobases upwards of the agouti gene. And I should say that the agouti gene is encodes the paracrine signaling, which has to do with the color of melanocytes and the color of the skin here, right? Or the fur, sorry. And the normal, that is recessive allele here, it has no insertion of the transposable element. If that transposable element is not methylated, it will cause a very yellow color. Whereas the not having it, it causes what black brownish, black brownish. If the transposable element is methylated, it's not so bright, it's more of a brownish type, right? So if we then feed this, we do a crossing here with agouti male in both cases and a female here, and that one is eating normal, and that is getting a supplement while pregnant. And the supplement is of the folic acid, which actually is methylation precursors, right? And also vitamin B12, I think, it's in there. And if you look at the progenies then, you will see that those getting supplemented, this should be about the ordinary one this type, and then you have the heterozygotes. They are heterozygotes and then in the same proportion and depending on how much is. And here you see there's much more of the brownish. Now, in these cases, you can go into and do a study of these genes. And what you find is that in this case, it's a little methylation in this transposable element here. Whereas in the cases where you get more brownish, there is a lot more of methylation. So evidently, this supplement does change the character here. And what is also to be noticed is actually that it promotes several disease phenotypes also, diabetes, obesity, and tumors. But that's another matter. But it is influenced by the environment, nutrition, and you will get a different pattern. And here comes a number of observations which came in around 2010 about that there were parental origin on some complex diseases which seem to have in basal carcinoma, type 2 diabetes, and there were some others also. So it had to do whether it was inherited via, or it seems that it was inherited via the mother or the father. And this was actually done on the, I'll take this one first, on the Decode. There was one on Decode, you heard of that? It's a company on Iceland, and as you know, Iceland has a population where you sort of can put them down into one huge pedigree to the large extent from the 30s, 14th century. And that's why they started in the 90s doing it. Anyway, it's called Decode, and they had looked at some of their parents to find that some of these diseases, common diseases, tends to turn up in families only when it is inherited from the mother or the father. Although in all other populations, and most of them, it comes from both parents. So it's not known that it's coming from, as you know, it's a complex disease. Anyway, this of course, as I said, this is came in the Times, the journal. And of course there was a lot of attention to it, and someone should make a little sketchy figure here of it. Unfortunately, that's wrong. And they didn't realize. Here's a mother, here's a daughter, and here's a granddaughter. Actually, what it says is that the daughter here, she should be influenced by the mother's essence. And but the granddaughter should be influenced by the, but that's not true. The germ cells of her baby here, the germ cells in the baby, are started here to be developed. That is the germ cells which will give rise to her daughter. The daughter's daughter, the granddaughter, right? It doesn't affect her. It does affect her granddaughter. Well, and there was this number of other papers around that time also showing that starvation during the Second World War was one of them. The women being pregnant when they were in these camps, and the grandchildren tend to have the common disease at a much higher degree than did the rest of the population. And we have in Sweden also areas where there was starvation in the beginning of the 20th century. And it seems also there though that the grandchildren there are getting, and there has been a number of reports in all other areas as well about it. So, a little bit about aging then. Here is monozygotes, twins. And of course we know that they have identical DNA. If you look at them in 3-year-old and when they are 50-year-old, and you use DNA which you have for one of the twins, you have prepared it with green fluorescence, and for the other with red fluorescence. And they use it as a CGH profiling, and you see they are mostly yellow, these chromosomes here. That means same dose you got from the twin 1 with green and the twin 2 with red. That produces a yellow color. Now when they are 50, you see it's quite different. The large extent here, there are a little bit which are yellows, but mostly there is more of the green or more of the red, depending on which is this. And that means that there are a demethylation and remethylation, probably mostly demethylation, since that is the case going with the aging. Anyway, it's also that when you are 50, you see much more discordance in the types of phenotypes, including diseases. One of the twins are getting the disease, the other is not. And and some studies have been done in looking in the particular cases, and you could see that sometimes it is also seen that it has to do with the methylation differences between the twins. Okay, so it's not. Now, what methods do we use for analyzing DNA methylation? Well, in general, what has been used and still, there's a candidate gene approach. That is, you have a given gene and you want to see if it is methylated or not. You use the methylation sensitive enzymes, so you bisulfite sequencing. And then there are a number of genome-wide DNA methylation profiling techniques, none of which is actually used anymore, so we don't go into any details about what is used. Is this bead chip phenium, and there are actually some others. I'm going into some examples, so that's why I'm writing that. Now let's first start with, there are enzymes which cut a CCGG, that is this recognition signal for this restriction enzyme. And if it is methylated, it is this C which is methylated, the CPG, right? The enzyme HPA2, it does not cut if there is a methylation, whereas the enzyme MSP1 do cut, whether it's methylated or not. So it cuts this in both cases. Right? You could use that for various means in various types of assays. What is mostly used in the bisulfite sequencing, and that's used a lot, including on this bead sequence analysis, it's that you treat the DNA with bisulfite. And we don't go into various steps, but a cytosine which is not methylated in the 5 position here, that will turn up as a uracil, which is in the RNA, which is equivalent to a T in the DNA. So, now this is we do in bisulfite sequencing. We don't have to go through the various methods. You get the sequencing, and when you get the signal, you go into into blast analysis. You put in as a query your own sequence, and then there is the compare in automatic, because it's available in the blast, the subject sequence for the same region, and you get the results out. It seems that there are 13 CPGs here, and 2 of them in your sequence were unmethylated, the rest were methylated. And you get it out, and you can show it on several examples for different genes and so forth. Right. I think we have to speed up a little bit now. So let's go into this bead and take an overview of the basics here. The bead chip, that is which Illumina has produced, and in various forms. It started for 2009 or something with the 27K. And then 2 years later or something, we had the 450K. What does it say? Yes, it says that these test the methylation status in 27,000 CPG sites in the genome. And this is 450,000 CPG sites. The principle of that is that there are beads and there is one sequence on each bead. And if you have a site here which is unmethylated, you have a bead here which carries the sequence in that particular site, and except for the second sequence in the CPG, so it goes in the middle there. So you have a unmethylated loop is bisulfite converted. So instead of having a C here, which you should have, if it was not bisulfite, there has been conversion to a U, which is a T when you do make a DNA out of it, which is PCR amplification. So then this B type is for the unmethylated type. So it has an A here, and then there could be a C which could couple to the G there. So you get a signal. Now, this same sequence here, if you attach it to the beads which has a G here, and this is a T here, of course, it cannot pair. And there could be no signal for the G. Yeah, right. So it's no signal. So for this one, you get a signal here on a bead which is known by the system that this is the unmethylated bead for that particular CPG. And here you have similar, where you have a methylated locus, or the same, by the way, one could be methylated and the other one unmethylated, of the 2 alleles you have in each gene, right? And you have in this, look at it here, it's fantastic. So it looks 12 samples you can have in here, This is for the 450, principally as I said the same. That means that there are 450,000 for unmethylated testing all number, and there are 450,000 to test the unmethylated and the methylated. This is in total almost 1 million beads in here. And it's small like this, right? Fantastic when you see it. Anyway, and these are very carefully, by the way, selected which CPG should be there, and it's continuously. Here's a first example of this. It's a clustering analysis of this is cancer. I think it was colon cancer. Yeah. That was when you used the 27K. Here you have the sample. Here you have the genes. In this case, 33,000 it was, or the CPGs associated with genes. Here, in the columns. And then you have the various samples here. If you just, in this region, I take a little bit over, you will see that in the top here, there are normals, and here you have colorectal tumors, and they are then, since it is only over this piece here, it's 2,500 or 2,250 genes. But anyway, no doubt. You could see clearly that there is a kind of profile typical for those having the colorectal cancer. Not in each and every one, but you see the overall picture that it is different if you have a tumor. Now the fifth, I shouldn't go into it now, but they have been very careful to what they are picking up, CPG islands. And actually they end up not in 450,000 but in 473,000 different sites. And actually I heard or read that the new ones I think is over 800,000 coming out now. If it is not. Anyway, I show another study we did 4 years ago or something. Had them off with that. Just looking at the obesity, DNA methylation in relation to BMI, I could say. We tested obese women and lean women. And what did we see? Well, we find that there were, if you're looking at those having the highest significance, they were not statistically proven, but at close, some of them were. But anyway, this was a small sample. We had about 10, 15 normals and about 10, 11 ones which were obese. They had the same age, and that's important, they were women. Because age, as I said, and you see clearly as well here. Typically, those which are obese, they provide a separate profile where they could be shown. Okay. Now, the last few years, except for producing a lot of this, and we should go further into that, I should say, when we discuss personalized medicine, genomics and personal medicine, next time. I will only mention here that you can and one has started doing also epigenome wide association studies. That is the equivalent to GWAS, right? Genome wide association studies. They use the 450 bead chips to select for the most significant differences that are on CPGs, and then they do an individual they do a GWAS study or use the same statistics, that is testing the individual site. If it is statistically significant that it is more methylated in, for example, those which have obese and not those who have a lower BMI. And in this was a small study, they ended up with only 3 which were significant, and they happens to be the genes here on chromosome 15-1 and chromosome 17 and 18. And you see them here in this picture. And there was a large study done coming out in the early this year. And I'll go through a little bit more of that because there's a lot of interesting aspects in them. And again, when we talk about personalized medicine next time. Anyway, in this case, they used over 5,000 individuals. They identified from Europeans and Eurasians, and they used a number of different tissues as well to look at it. And they ended up with 180 or 90 or something. I forgot the figure, I shouldn't be on that, but I forgot. Anyway, they show the same here, an eBay study of these 189, I think. But to get them on the picture, they did it here as a circus plot, they say. So there is a number of them, and they've seen, and as you mentioned it already now, that they were significant differences in obese compared to normal, found both in obese tissue, but also in blood. Most of them also can be seen in blood samples, which is interesting, and you saw them in some other tissues as well. So, lastly now, I just mentioned that there are a number, actually 3 large major epigenome projects, and they are rapidly increasing, as you say. And the NIH roadmap, Epigenomics roadmap, is a human epigenome project, which is mostly European related, and you have the International Human Epigenome Consortium, and you have the International Cancer Genome Consortium. And they are devoted into various aspects, I should say. The roadmap, Epigenomics, and NIH, it has taken a specific interest in epigenomic mapping of stem cells and primary ex vivo tissues. Whereas it comes to the Epigenome Atlas, it's sort of specifying or cataloging and have reference available for modifications of specific locus, specific cell types, and so forth. When it comes to the Epigenome project, it's more related to DNA methylation patterns in human across the world. They are looking for about, about 1,000 people, representing various populations in the world, and analyze and catalog typical epigenome patterns. And the human epigenome, they map and establish functional significance of DNA methylation. I mixed it up. So when it comes to the IHEC, that's the one who are cataloging epigenomes in 1,000 different 1,000 individuals in different populations of the world. And they say that they will include 200 different tissues, which sounds very much, but if they are lucky, they probably will get a large extent of the management. And then, of course, you have the International Cancer Genome, and that's a large one. There's a lot of cancer, of course, and they are doing a lot of transcriptomic, genomic, and epigenomic, 50 different tumor types, and also subtypes of them, which are the most common ones in various parts of the world. Okay, so a conclusion here is the rapidly increasing epigenome technologies. Indeed, it is. And then the information which comes up increasingly more in the International Genome Project does no doubt indicate that the epigenomics is in the threshold to a new era, which will, by which we can understand the human development in health and disease and the impact of environmental factors, not least, on gene expression. And non-DNA based knowledge will have a major impact, and there has been a lot of discussions of how in finding new avenues in preventing and in diagnosing major human diseases. A little bit of that we will come into further next time we meet. So, thank you for today.
Yeah, could I ask you this erasure process that goes on once the zygote is
You think in the early development?
Yeah.
Yeah, right. To the blastocyst. Yeah.
and up until blastocyst time.
Yeah.
What happens if, I mean, there has to be a mechanism to do that.
Yes.
So
Yes.
what happens if that does not
Work.
occur?
Yeah.
Or doesn't occur completely? So it's,
I guess it's the same as with if you have chromosomal aberrations. You know that you have during development there is at each stage sort of a testing of phenotypes. If it doesn't work, you will get the embryo will die or the
lose the
Yeah,
embryo.
you will lose the embryo, exactly. And I think, I don't know, some people have probably looked at it, but I think you would have exactly the same if there are methylation differences. You know, if you have 3 chromosomes of any except for the X, that will lead to that the embryo will be out, right? It doesn't work. And that goes for all others. So the figure I have, although I'm not in the business, you know it much better, I think that is it 40 or 30% of all zygotes which actually develops into a child, right? The rest is during the various steps of testing, which is a very smart way, as we think of it, that test that is everything okay when you come to the various stages of the development. If not, okay, it's no idea that it continues, well. At least the way I look at it. I might see it otherwise. Yeah. Well, there is a lot here, and it's of great importance. I will be available if you would like to. Oh, you had a question?
I have a quick question.
Yep.
I'm just wondering, I think this maybe gets back to Dr. Skinner's question about marks that make it through that eraser period. There's some residual methylation that does not get erased, and I'm wondering if that leads to heritable disease. And there's also this possibility that you may restore marks that lead to disease.
Yeah.
And I'm just curious if there's much known about that.
No, it isn't, but it's known as that there are these. To what extent they are of importance, that's known. But there are cases of various changes, either that the is not demethylated as it should be, certain is still there, or that the methylation is not coming up as it should in the next division or whatever. You know, this maintenance methylation system, the DMT1, it means that cell division after cell replication of the genome over and over again keep by the system in principle also the methylation status of each CPG exactly the same. But nothing is perfect, so now and then, as I said, it doesn't work and that it leaves a change. And that goes all the way. So, but there is a lot of specifics to that, I'm sure, which we don't know about.
Can I ask a sort of similar, it's not a similar question, but these are questions we've sort of been asking for a long time. But the question that I have that was based on something that I read a long time ago that I could never find is that brain tissue has the highest level of methylation. And if that's true, it sort of argues that brain is more tightly regulated from a transcriptional.
It's what? It's
more tightly regulated, so there's less noise, transcriptional noise in the brain than any other tissue, which would probably be, you'd make sense of that, but with all the encode stuff and everything, do you know anything that?
Yeah, I heard that already. That was quite some time ago. After that, there's a lot. Then there has also been speculation. I said that 10 to 20% of the genes, at the most 10%, 20% of the genes are active in any given cell type. But there are people doing some studies about brain cells, in certain brain cells, where it might be up to 25 and they even claim 28%, I think, in one article. So that there actually should be more active genes in brain than are in other tissues of the body. And that also takes, of course, it's more active, even more complex control situations so that it works when it should and to the extent and so forth. Right? But it is interesting, of course, but it seems that due to the complexity and not also the brain is not easy to analyze at the molecular level, of course. There is particular studies doing it. So I don't know the answer to it, but I know that there is. There could be actually that there are more methylation in the brain or that there are more genes active in the brain than in other tissues.
Yeah, no, I wasn't suggesting that there's less genes active, just that it's more.
The methylation or the epigenetic processes are therefore more precisely regulating the genes. Yeah, I understand it. There is studies doing that, I'm sure. I know. And Well, we'll see. That's one of the areas which we have to work a lot on in order to understand more precisely, I guess. But of course, it's important. Yeah. No questions now. I am available, you know. So if something turns up, you're welcome to contact me. I'm sitting in the room. Thank you.